This is going to sound crazy, but cancer is a good thing for the survival of our species. And I'll explain all that later.
Most people would laugh, move on, choose something realistic. He didn't. Instead, he became obsessed with a question that almost no one at the time was asking seriously. Why do we age at all?
I was only 10 years old, but I'd already started saying to myself, I don't like the fact that I'm temporary.
And that question took him deep into the machinery of human biology, into the parts of our cells that quietly determine how long they live and when they die.
People don't like me saying that aging can be cured.
If aging is something we can influence, what happens next?
And I'm sitting there while he's talking and I'm thinking, oh my God, this doesn't just explain the limit on our lifespan. Because what he was saying is that every time our cells divide, our telomeres get a little shorter, and that when we run out of telomeres, essentially run out of ride tickets, our cells can't divide anymore and we die of old age.
Today's guest has spent his entire life sitting at the center of that question. A pioneer in genetics, a key figure behind the discovery of human telomerase, and someone still pursuing what many consider impossible, challenging the way humans age. This is the story of Dr. Bill Andrews.
I just want to cure aging. That is my goal in life. I have no interest in becoming a celebrity or anything like that.
You're also known as an ultramarathon runner who competes in extreme endurance races. So how has studying aging influenced the way you personally live your life?
Are the research labs focused on trying to cure diseases? No. They're focused on trying to get publications. 99% of all scientific publications are garbage, okay? They're publishing something that everybody already knew.
Bill, welcome to Drug Diaries. Before he ever stepped into a lab, before the discoveries, the patents, and the decades of research, he was just a kid, being told something most people would dismiss instantly. You should grow up and cure aging. Most people would laugh, move on, choose something realistic. He didn't. Instead, he became obsessed with a question that almost no one at the time was asking seriously. Why do we age at all? And that question took him deep into the machinery of human biology, into the parts of our cells that quietly determine how long they live and when they die. And years later, after relentless work, failed attempts, and scientific resistance, he helped uncover something extraordinary. A mechanism, an enzyme, a potential key to one of humanity's oldest problems. But here's where the story takes a turn. Because discovering something like that doesn't automatically change the world. It challenges it. It creates tension between science and skepticism, between innovation and regulation, between what's possible and what's allowed. And it raises the question that becomes impossible to ignore. If aging is something we can influence, what happens next? Today's guest has spent his entire life sitting at the center of that question. A pioneer in genetics, a key figure behind the discovery of human telomerase, and someone still pursuing what many consider impossible, challenging the way humans age. This is the story of Dr. Bill Andrews. Bill, welcome to Drug Diaries.
Thank you very much. Let me just comment on what you just said. You know, I just want to cure aging, okay? That is my goal in life. I have no interest in becoming a celebrity or anything like that. I just want to cure aging. But the reason why I want to do interviews like this is because I want the world to know that there's some really major problems when we can cure cancer and heart disease and things like that, but there's obstacles in the way that people are not aware of. And that's one of the reasons I want to make it clear so that after I do cure aging and they do make a movie about it, and in that movie, I'm going to make a really big goal of making this point, is that I want other diseases to be cured without the scientists going through the same obstacles that I have. And it is unbelievable, the amount of obstacles. When the science is clear cut, we know exactly what to do. It's just, there's sometime during this episode, I'll get a chance, I'm sure, to talk more about all the obstacles.
Absolutely. Absolutely. And Bill, you've spent your life trying to solve one of the humanity's oldest problems, aging itself. And before we get into the science, I would love to start with the story of how that mission began. And I read it somewhere. Your father once told you as a child that you should grow up and find a cure for aging. Do you remember that moment? And did it actually shape the direction of your life?
I remember that moment better than you could imagine. I mean, I know exactly where I was standing. I know exactly what I was doing. I mean, it was a, what do you call it? An aha moment in my life. I mean, it's okay. So I had already, I was only 10 years old, uh, but I'd already started saying to myself, I don't like the fact that I'm temporary. Okay. I don't like the idea that only going to exist for a little while and then be gone. And I didn't like the idea that the same was going to happen with other people that I knew and loved and things like that. I just in school, in elementary school, back then, this is, I'm talking about the early mid 1950s. Okay. Back then, we didn't have cell phones. We didn't have, we only had like a single phone in the house if we're lucky, hard line. There was no such things as answering machines. I didn't have my first answering machine until I was like in my 20s. And I thought, wow, what's the greatest invention of all time? So communication was all by letters. You wrote a letter, you put a stamp on it, put an envelope, put a stamp on it, sent it. But teachers and other things, they had better ways. And that was what got part of this ball rolling, is that my elementary school teacher, and one in particular that I remember very well, except I can't remember her name, she was really into science and medicine and talking about things like that, us doing science projects. And I was like blown away by all of it. And I was asking all the questions that no 10-year-old should be asking. And she one time pinned a note to my shirt and said, when you get home, ask your parents to read this note. And the note said, your son is very interested in science and medicine. You should nurture this. And so my parents already kind of had a clue, because I told them that for Christmas, I wanted a telescope. Okay. And so on Christmas Day, I open up the box and it's a toy telescope. And literally, I cried, okay, because I didn't want it. I wanted a real McCoy. And so
Real telescope.
my parents were so upset about it that they put me into a car and drove us around to yard sales. It was right after Christmas. And, we found this big 4-foot long, 8-inch wide reflector telescope that had all the gears and everything like that to track the stars and stuff like that. And it was like no 10-year-old kid would ever want anything like this, but I did. And my parents ended up buying that, and we took it home, and I was just addicted to it. I was on it every day, looking at the stars, every night at least, looking at the stars, finding, I discovered Saturn and its rings, and I discovered Jupiter and its moons all by myself, just by looking at one star after another. So that note I came home with that note. And what I remember is I was out on the front lawn at the, I want to say the southwest corner of the lawn with my telescope looking at stuff. And my father comes out of the front door of the house and he walks diagonally across the lawn. This is how much detail I remember. And he walks up to me and he says, Bill, since you're so interested in science and medicine, when you grow up, you should become a doctor and find a cure for aging. And then he said, I don't know why nobody's done that yet. And so right away, I said to myself, oh, that's a great idea. I would love to do that. And it's like, I already was upset about the fact that I had learned that life is just temporary and things. And, and I wanted to, everything I wanted to do, I wanted to find a way of making myself invincible. Okay. And not just me, but other people too. And so I thought this was a great idea, and it just stuck with me so much. That was a really big moment in my life, and that's why I remember it so well. But my father and I became partners, especially when I was in college. When I started really taking this on when I was in college, and he and I would talk about curing aging all the time. And by the way, I didn't find this out until I got even out of graduate school. People don't like me saying that aging can be cured. Okay. It doesn't make sense. It's a concept that they don't understand. It's like they never thought of aging as a disease. And I always did. I don't know why nobody does it. Because even when I was 10 years old, I thought aging has to be something wrong with us that can be fixed. And And I just, at the time when I was 10 years old, of course, I didn't know anything about DNA or I knew about cells, and I knew that they can divide, but I didn't know, and I knew we were made up of cells, but I didn't know much about anything. But I just thought, okay, so I've got to start learning. I gotta focus my education on learning everything I know to be able to eventually cure aging. And I refer to it a lot as I had to build an arsenal of tools. Okay. But, my father and I would talk about this all the time. Even when I was in college, we'd get on the phone and talk about it, talk about curing aging. I'm going to skip. My father ended up passing away in 2015 from Alzheimer's disease. And I'm
Oh,
sure we'll come
sorry
back
to
to
hear.
more on that. But that was a devastating experience for me because I lost my best partner in this whole endeavor. But it's like, the frustrations. Why didn't I cure aging before he passed away? And the frustrations there. And the same with my mother. Why didn't I cure aging before then? Okay. When I thought, God, it must be really easy. But we'll spend some time on the obstacles later. But
Sure, sure.
the world needs to know that it's not as straightforward as everybody thinks it is.
Yeah.
Yeah, but yeah, long answer to a short question. I do remember that moment. I will never forget it. And if people watch the documentary called The Immortalists,
Mm,
they'll see my father in there. And we both talk about
I saw him.
that
Yeah.
day.
Yeah. And Bill, when you look back at your younger self entering molecular genetics, did you already believe aging was a solvable problem?
I mean, I don't know why, but I thought it has to be some kind of disease. It had to be something we fixed. I mean, I can't remember if I really thought this when I was 10 years old, but it was not too long afterwards that I started saying to myself, well, if we have cell division, why can't we repair all the damage? Because, okay, so that was one thing. When I was 10 years old, I thought aging was totally
Mm,
due to wear and tear, totally due to exposure to the sun and the wind and the rain and things like that.
yeah.
And that was the only reason we aged. And I thought, but we have cell division. Can't we just, and this is what that teacher told me. And when I was 10, this teacher that
Mm,
was really into, she told me that our cells divide and repaired things and things like that. And I didn't just let that go in one ear and out the other. I thought that's a very important thing to know. And so, but it bothered me that why don't we repair all of our damage and just live forever? But that's what I was doing at the time. So I thought, yeah, I thought aging could be cured, and I thought all the diseases could be cured, and I thought that would be a good use of my time. That's
yeah.
exactly what I thought. So
Right. And there's this fascinating moment in your career when you attended a conference, anti-aging conference, and heard about telomeres and telomerase. What was that moment when you realized this might be the key to aging itself?
you know, that wasn't my first aha moment. I mean, there were other things that happened along the way, but let's talk about that one, and then let's come back to
Mm,
the other things, the big aha moments that occurred before then. But the I was sitting in the audience, and I knew certain things that I needed to find out to explain some what kind of like explanations that I had for why we age. I just needed to put the pieces together. And one obstacle was, and I'll talk about this later, I decided there had to be something like ride tickets in an
mm,
amusement park. And I know that sounds like a silly way of saying it, but that's exactly what there had to be something like ride tickets in an amusement park that limited the number of times our cells could divide to repair our damage. And that would be the only explanation as to why we don't repair our damage all the time. And so what was the biochemical mechanism for this? And after I got my PhD, I I didn't go into anti-aging research right away because I still had to do the deep thinking to try to figure out what those ride tickets were. And so I went in and did other things. We'll come back to that too. But
mhm.
I'm sitting in this audience at an anti-aging conference with my father, by the way. He would go to
Okay.
these conferences with me. He was a TV producer. He wanted to make a documentary on cure for aging. This is how obsessed both of us were on the subject. So I was introducing him to all the world leaders in anti-aging. But I'm sitting there, and this guy named Calvin Harley, Dr. Calvin Harley, gets on stage and he starts talking about how telomeres shorten.
Mm,
And I'm sitting there and I'm thinking, oh my God, here's those ride tickets. There they are. And I'm sitting there, and my whole career up to that point has been all about uh, recombinant DNA research, genetic engineering, expression, turning genes on and off, gene expression and stuff like that. And I'm sitting there while he's talking, and I'm thinking, my God, this doesn't just explain the limit on our lifespan. Because what he was saying is that every time our cells divide, our telomeres get a little shorter. And that when we run out of telomeres, essentially run out of ride tickets, our cells can't divide anymore and we die of old age. But I thought to myself, that's a limit on our lifespan. So he was explaining the limit on our lifespan. But I, with my background in gene expression already, was sitting there thinking, my God, these have all the hallmarks of what are called enhancer sequences. And I thought, not only is the telomere shortening affecting our limit on our lifespan, but it's also affecting all the changes that we see in all of us as we age. The synchrony. There's a synchrony. It's like the analogies that a lot of people use is, why does our left arm age at the same rate as our right arm? But more importantly, why do we see, when we walk down the street and we can see people, we can estimate their age and know what it is because of the synchrony in aging. We all age the same way. Humans are one way, but dogs and cats another. They age entirely differently. And that was another clue as to what's going on. But I knew that there had to be something that was regulating our genes, gene expression, so called the epigenetics, are some genes. So when already I had done experiments where I had looked at gene expressions of young cells and old cells, and I knew there was a profile, there was a change that occurs in everybody. But how did those changes come about? What caused those changes? And it can't be just the environment, because the people in the North Pole and the South Pole were aging the same way people who live on the equator were. And dogs and cats are aging at different rates than we are. So it couldn't be just environment when the dogs and cats are in the same environment we are. And so, but I'm sitting there thinking, oh my God, these telomeres are acting as enhancer sequences. They're actually folding over like enhancer sequences do, coming in contact with genes, turning them on or off. And when I talk about turning genes on and off, I talk about how every gene has a dimmer switch next to it. And so it was regulating these dimmer switches. And the difference between telomeres and enhancers were that telomeres got shorter. And the shorter they got, the less capable they were of being able to reach the genes. Now, I remember, so I'll come back to that in a second, but I went right up to the podium after this guy's talk, before he even got off the stage. And I said, has anybody figured out how to re-lengthen telomeres? And he said, no. And he said that we've been working with teams all over the world, our own labs and things like that, and nobody has been able to figure this out yet. And I just flat out said to him, let me come and work with you and I'll have it figured out in 3 months. And
That's a very famous quote.
it sounds kind of crazy, but I already had this absolute unbelievable reputation of being responsible for solving the major problems in practically every biotech blockbuster breakthrough that had occurred up to that time, including human growth hormone, tissue plasminogen activator, erythropoietin, beta serum. There's all these things that I had become known as the ace of. Some companies like Genentech and Amgen were contracting my company to be able to get access to me, my company being the company I worked for at the time, get access to me. My boss, who was Joe Hedgepeth, he was like all over this idea. He thought I was a wonder child, okay, in terms of being able to make things happen. And so he would get all these things going. And I became the ace. So Calvin Harley, who coincidentally ended up having worked with Joe Hedgepeth years earlier, had already known about me. So when I said to him, I'll have it figured out in 3 months, he did not doubt it at all. And he hired me essentially on the spot. Shortest job interview ever. I was a lifer. I said I was never going to leave the company I was working with, which was Codon Corporation, who changed its name to Burlex Biosciences. But I left them in a second to go work with Calvin Harley on trying to solve this aging problem by figuring out how to re-lengthen telomeres. And 3 months later, my team, he put 3/4 of the company reporting to me, which was about 30 scientists. And I remember on December 7th, my very first day there, December 7th, 1993, we had this major historic strategy meeting where we outlined about 10 different ways we were going to go and figure this out. And I assigned different scientists to be in charge of each thing. And then I was actually in the lab myself working on one of the projects. But 3 months later, I announced we had discovered human telomerase. And
No way.
the key was we realized that if every time human cells divided and telomeres got shorter, how could children be born with longer telomeres than we have? When the production of sperm and eggs comes from cell division of what's called primordial germ cells. So there had to be something in those primordial germ cells that was preventing telomeres from shortening. So that was our target, and we discovered this enzyme, human telomerase. I say human telomerase because the enzyme had been discovered like 10 years earlier in a pond scum organism called tetrahymena. But they never connected the dots to aging and cancer and stuff like that. But Calvin Harley, he was the one who proposed the idea that this might have something to do with the aging process. So he was taking this on, but he didn't have the scientific skills and stuff like that to make it happen. And he was frustrated with everybody he was bringing in on the project. They couldn't get it done either. So he brought me in. And 3 months later, but the fact that we found it in our reproductive cells told me that we have a gene that's producing this enzyme. And if our reproductive cells have it, all of the cells of our body have it. It's just shut off for some reason. And so that was the next 30 years of my life has all been figuring out how to turn that gene back on. And in all of our cells, not just our reproductive cells. And you wouldn't believe the obstacles I've had to go through to continue this thing. So let me just, I know we're going off, but one of the biggest obstacles was people telling me, oh, that's just going to cause cells to become cancer. Okay, well, all these people that are saying this were not cancer experts at all. I had no background in cancer. But I was already National Inventor of the Year in the United States for my cancer research, but for a lot of the stuff I had done at my previous companies. So I knew a lot about cancer, and I knew that was nonsense, but I couldn't believe the amount of obstacles I had, the amount of complaints that I had from other scientists very publicly about turning telomerase on in all of our cells is only going to give us cancer. And I kept saying, no, it's not. It's going to decrease the risk of cancer. But I don't want to get onto that. I'm sure we'll come back to that whole subject. But, that was a big day. So we demonstrated that we could actually stop the aging process in human cells in a petri dish. And that was headline news. We're really, a lot of people claim to stop aging, but we were the only people that have ever been able to do that. And so far, turning telomerase on in normal human cells, or what we were doing, was putting a new gene in by gene therapy, because we still didn't know how the gene was regulated. So we were just putting this new gene in. And we showed that we stopped aging and reversed aging in every way imaginable that you can test in a human cell in a petri dish. I'm sorry to go off on tangents.
No, this is all music to my ears. It's so fascinating, and I've got so many questions. And
Yeah.
if you were to take a step back and go to the basics of science, and for someone hearing the word telomere for the first time, how would you describe it?
Well, ride tickets at an amusement park would be the best way to describe it. But they are found at the very tips of our chromosomes. One analogy that is often used is they are like the aglets on our shoelaces. Okay, think of a shoelace as a chromosome, and along the shoelace is all of our genes that give us our hair color, our eye color, things like that. And all these genes are turned on and off by dimmer switches called promoters. And so, but at the very tips of your shoelaces are these caps that protect your shoelace. They're called aglets. Well, telomeres are like those. Now, I know a lot more about telomeres than just that, and so I know they're not exactly the same thing. They do protect our chromosomes from damage. They do play that role, but they don't degrade. They don't unravel. They don't, all the words that everybody uses to explain why telomeres get shortened. That's not how telomeres shorten. You can cause them to shorten by wear and tear processes like damage to the DNA, things like that. But you have to really have a bad choice of lifestyle and things like that, and diets to doing that, because the real reason why the telomeres shorten is because every time our cells divide, the DNA, the chromosomes need to be duplicated. And when the DNA is duplicated, called DNA replication, it can't replicate all the way to the very end. It's called the DNA,
Oh,
the
I see.
end replication problem. That's an actual scientific name for this. It's called the end replication problem, whereas the cell cannot copy the old DNA to make the new DNA and can't copy all the way to the very end. I explain that like a bricklayer making a new row of bricks on top of a brick wall, and the bricklayer is standing on top of the wall, stepping backward, laying a brick that's in front of him, then stepping backward, laying another brick. And then when he or she gets to the end of the wall, that bricklayer falls off the wall, and the last brick was never placed. So the new row of bricks is shorter. Then the next row of bricks is shorter than that, and the next row of bricks is shorter than that. That's how, so it's not a degradation problem. It's actually the lack of the ability of the cell to replicate to the very end. And so that's why every time a cell divides, the new chromosomes are shorter. Okay, so that's equivalent to losing a ride ticket, and that's what's happening.
Bill, you led the research that identified the RNA and protein components of human telomerase. Why was this discovery such a breakthrough in biology?
Well, because everybody would say that when, the fact that human cells can only divide a limited number of times, that's called the Hayflick limit. And that was I said there was previous aha moments. Well,
Yes.
back in college in the 1970s, when I first heard about the Hayflick limit, I got all excited because that explained to me why we can't repair our wear and tear indefinitely. And that would explain why we have a limit on our lifespan. So that was an aha moment, because up until then, I just couldn't make sense, but I didn't understand, why do cells only have a limit on our lifespan? But so that's where the telomere stuff came in. But it was like everybody, the whole attitude was that this whole thing, when the cell reaches the Hayflick limit, it goes into a phase called senescence. And it was even taught in school that senescence is completely unreversible. You cannot avoid it, you cannot prevent it, and you can't reverse it. It's impossible. And, telling me something's impossible is a great way to make me go after it. And that's always been the case. If we get back to that, I'll tell you about a whole bunch of things I've done that when people told me something was impossible, it made me go after it and show that it was possible. But I was bothered by the fact that senescence was something that was said to be unavoidable. So when we discovered telomerase and put it into cells and showed that the cells did not go into senescence, and then later I was able to show, after I started my present company, Sierra Sciences, I was able to show that even when we had senescent cells, we could turn the gene on and the cells would come out of senescence. Okay. And so we could not only stop it, prevent it, we could reverse it. And so that was why that was such an important thing. Now, when we announced it, we only showed that we could prevent it at that time, and that made headline news. That was earth-shattering, because it destroyed a viewpoint that everybody in the world had, and suddenly that wasn't true anymore. And so, but unfortunately, everybody said, well, you're turning the cells into cancer cells, because we knew that cancer cells don't have a Hayflick limit. They just divide indefinitely. And but this is such a nonsense world. Cancer is the lack of growth control. Yes, cells can divide and divide and divide, but they only become cancer when they divide uncontrollably. That's why our noses don't grow longer than they have, because something in our cells communicating with cell-to-cell interactions was telling our nose, stop growing. Okay, you've reached your point. Don't divide anymore. Well, cancer is when all of a sudden the cells ignore those growth controls and they start dividing. But telomerase, lengthening telomeres, was not turning off that control. It was only providing the cells with more opportunity to divide when they needed to divide. But whatever. So going another tangent there, but I'll come back.
And that is where I'm going to dive into the science with my little knowledge, and correct me if I'm going wrong and pardon my ignorance. But I believe that When the cells are exposed to certain radiations or chemicals, they almost have a choice to either become cancerous or become senescent. And when they become senescent, they're trying to prevent the formation of cancerous cells so that they don't duplicate and they don't multiply the formation of cancerous cells and they turn into zombie cells. So you are saying that, is that understanding correct or
Yeah,
am I
well,
wrong?
okay, so one of the really, really big problems in biology and curing diseases is the whole idea that cells tried to do something. Okay, and
Mm,
that's what, when I was in graduate school, my biggest focus there was to try to understand evolution. Why did we evolve things? And there was no, evolution is not a force, it's a result. Okay, and we'll spend a lot of time talking about evolution later too, I'm sure. But it's like our cells didn't develop this senescent process to
mm.
prevent cancer. And that's a lot of people think that.
Yeah, I
And,
was under that impression.
but actually, and this is going to sound crazy, but cancer is a good thing for the survival of our species. And I'll explain all that later.
How's that?
Okay. It's so we would never, ever evolve a way not to get cancer. We would only evolve ways to regulate cancer better to make sure that it only affects, in most cases, the longer lived people, not the shorter lived people. And by longer lived, I mean, once you've lived long enough to raise your young, you now are considered longer lived. And the sooner you're eliminated, the better the chances of the species surviving. And I can explain that. People get impatient. They can always go to my website and click on a link called Key Videos and Documentaries. And I spend up to an hour sometimes just explaining the whole subject of why we age and why cancer, heart disease, Alzheimer's, things like that are important for the survival of the species. And that's why we never evolved a way not to have them. But it's like, uh, there's been a lot of evolutionary theorists in the past, but I don't think anybody has ever been working on it as long as I have. And I'm not considered an evolutionary expert, but I think I know more about evolution and the what's and whys and hows and everything like that than anybody on the planet. I mean, I'm a modest person, but I'll say something like that because every time I talk to anybody about evolution, I can easily convince them that they're wrong. And when they listen to what I have to say, they say, "Oh my God, I hadn't thought of it that way." And so, well, whatever.
Yeah.
I'm just going
And
tangent after tangent.
no, but that is so fascinating. And what you just said about cancer being important and critical for our survival really stopped me in my tracks, and I'm still not able to wrap my head around it. So, explain to me a little bit more about why do you think so, and why do you think cancer is, in a way, good for our evolution and survival?
Well, because let me think about a good way to phrase this. When there's like rapidly changing things in our environment, the species has to be able to have some way of adapting to that. Otherwise, it'll go extinct. And adaptation is a
Mm,
bad word, okay? Because we don't adapt. Adapt's a force. It's instead of adapting, the species just hopes that it has some members of the species that have a particular set of traits that will allow it to survive that
mm,
rapidly changing environment. And let me just say, when I say rapidly changing environment, I don't mean just asteroids hitting the Earth. I mean new viruses and bacteria that infect us. I mean new predators. I mean environmental changes too. That the concentration of like oxygen in the air changes. All kinds of environmental things can happen. But the way to survive them for the species is to just have diversity within the species. And throughout evolution, every step, every major step in evolution has been to, I don't want to make it sound like it was a plan, but the
mm.
result of every change was that it increased diversity within species. I mean,
Yeah.
mutations happen
Mutation.
right away. Mutations increase diversity. And then there was also this, I'm forgetting what it's called, but there's like viruses and things like that infecting us, causing new changes in our cells. That also caused diversity. But one of the biggest changes ever in diversity was sexual reproduction. Okay, and that I think that occurred about 2 billion years ago. Up until then, it was all asexual reproduction, and so the progeny would be identical to the parents, and so there wasn't very much diversity increased, and so species were going extinct left and right. But sexual reproduction now all of a sudden started shuffling our genes. The male mates with the female, the cells come together, they have different genetics already. Now they have this new combination of shuffled genes between the two parents and create an offspring that never existed before. Okay, very totally different from asexual reproduction. And so that was one of the greatest things that ever happened in terms of evolution and survival of species. I mean, individual species that had sexual reproduction were much more likely to survive rapidly changing environments because of this increased diversity. But the asexual reproduction organisms still survived because there were so many of them. Okay. It wasn't a survival of each species, it was survival of at least one of those species that survived, that bred asexually, and that was how it worked. But now all of a sudden it increased the survival of each particular species to survive by having diversity within the species. Okay, so almost immediately after the evolution of sexual reproduction, we evolved aging. Okay, aging did not exist before then. And the reason why we evolved aging, not because it was a force or anything like that, because the species that ended up accidentally having ways to eliminate the longer lived that already had raised their young, had an accidental way of eliminating the longer lived, had a better chance of surviving because there was more diversity added by allowing the offspring to interbreed as opposed to the parents rebreeding or even breeding with their offspring in such cases. Okay, and so just those species that had ways of eliminating the longer lived were more likely to survive the rapidly changing environments. And so what was important, it wasn't just anything unhealthy that increased the species, it was things that didn't become unhealthy until you had raised your young, at least in most cases. I mean, cancer is one of those. Okay, cancer affects the longer lived a lot more than the shorter lived. There are people, children, that get cancer. So it's not perfect, but it's still, the way evolution has worked is it still eliminates the longer lived from cancer more than the shorter lived. And so it increases diversity within our species and increases our species' ability to survive rapidly changing environments. But the same, it's like when you look at all the diseases on the planet, you start realizing that most of them affect us after we've raised our young. And so, back in the old days, in ancient days, we typically had children when we were 13 years old. Okay. And then a human could survive on its own, hunt and everything like that, by the time it was like 7 years old. Okay. Yeah. And so, I mean, it seems unimaginable now, but that's the way things were back then. And so we became longer lived by the time we were 13 plus 7, 20. Okay. And a lot of people will say, well, there's still an advantage because grandparents help raise your children. Okay. So that may or may not be true. So instead of 20, which is 13 plus 7 is 20, let's make it 27. Okay. I mean, it's like we reached an age of being longer lived at a very young age. And so our average lifespan, even in the 1900s, was only like 40 years old. Okay. And so everything that, so we are intelligent beings. Okay. Our species survival is no longer dependent upon evolution increase in diversity. We can control our own evolution or our own survival now. And we've done a terrible job. I mean, a lot of species are now extinct because of human intervention. Okay, so there's a lot of dumb things we've done, but that doesn't mean that we don't have the ability to control our own evolution. And that's what I'm doing. Okay. I'm focusing on trying to undo all these diseases that evolution gave us. But the way I often put it, I said, I'm trying to undo the things where evolution failed us. Okay. It failed us as individuals, but it didn't fail us as a species. Okay. But now I can undo those, and other scientists can undo those kind of things. And that's where I want to
Yeah.
get to eventually someday, is talking about the obstacles to being able to do that.
Yeah, and this reminds me of my recent conversation with Dylan Livingston, and we were talking about how rapidly the aging and longevity tech is evolving, and we are close to immortality. And on the other hand, we've got the AI revolution, which is going to make us all redundant, and nobody is going to have a job in future. And the birth rate is going down rapidly. In a way, we are all turning into senescent cells.
Did you say David Livingston or Dylan Livingston?
Dylan Livingston.
Okay, yeah, I know who Dylan is. I'm a great fan of his stuff.
Yeah, he's
And
amazing. He's a
we'll
great
we'll
guy.
have to come back and talk about some of the stuff where my work overlaps with his. Track of how did you start the tangent?
Exactly. So let me go back to the topic of telomerase. And so, what did the scientific community initially think about the idea that activating telomerase might extend lifespan?
I mean, a lot of people said aging can't be that simple. Okay, and maybe it's not. Maybe we might have to do more than that. Okay,
Hmm.
the analogy that I used when I try to debate, I don't like to debate, even though I'm really, really good at debate. I've taken a lot of courses in debating, but I've only learned that debating doesn't reveal the truth. It only reveals who's the best debater. And so I don't like to get into debating, but I would debate them by saying, we have multiple sticks of dynamite inside of our
Hmm.
cells, and they're all burning. They all are affecting our aging. They're all burning at the same time. But we have to focus on which stick of dynamite has the shortest fuse, and we need to put that fuse out. And after we find out what that fuse is, then we ask, well, are there other sticks of dynamites burning too? And then we need to work on those. But this is all the process of living long enough to live forever. Okay.
Hmm.
I forget his name suddenly. Doctor that coined that phrase, that's a good friend of mine, Dr. Terry Grossman. He
Right.
coined that phrase. He published a book with Ray Kurzweil on that. So we have to put that first stick of dynamite, the fuse on that first stick of dynamite out. And maybe that might end up adding another 20 years to our life, another 200 years to our life. We don't know. We have to find out as we go. Okay, so I'm 100% convinced that in humans, also dogs, cats, horses, sheep, pig, and deer, and they're the only ones, and also non-human primates, our shortest fuse is telomeres. Okay,
Yeah.
telomere shortening. We have to put out that fuse. And then we might find out that we have other fuses burning, like mice. Mice
Hmm.
and rodents and rats, or all rodents, mice, rabbits, rats, their shortest fuse is not telomere shortening. Even though a lot of
Oh.
people are studying mice and looking at telomere biology and find out that they can extend lifespan a little bit in mice. I mean, extending a little bit is not the goal. We want to totally stop aging. Mice just have these other mechanisms that's causing aging, most notably oxidative stress and mitochondrial
Hmm.
dysfunction.
Yeah.
But humans have 100 times the amount of natural antioxidants than rodents do. And that was published mostly by Dr. Richard Cutler years ago, where he looked at antioxidant levels in humans versus mice, and he found that the humans have 100 times as much. So mice suffer tremendously from oxidative stress and mitochondrial dysfunction, and that also results in a very rapid telomere shortening when they get older, but the telomere biology is extremely different. Mice already have telomeres produced in all their cells, okay,
Hmm.
and they have telomeres sometimes so long we can't even measure them. But it's the oxidative stress and mitochondria function that's causing their decline. And maybe after we solve our telomere shortening problem, and when I say we, I mean hopefully I
Yeah.
do, but I would love it if somebody else did, okay, because I want them to cure my aging. But that's going to give us a number of years longer to work on putting out the other fuses. And maybe we might find out that we do have oxidative stress or mitochondrial dysfunction or something like that, killing us off when we're 200 years old. Okay. And if that's true, that gives me another 80 years to solve that problem. And believe me, I'm not stopping until I'm immortal. It sounds nuts when I say something like that, because we're obviously won't be immortal because we do dangerous things that kill us all the time.
Mm.
They have nothing to do with aging. But it's like, I want to see the decline of health disappear from aging. And we'll go into that a lot too. I just think that if we are serious, we can actually get ourselves better and better. And then there's also things like mind uploading, or it's called whole brain emulation or something like that. That is actually going to make it so that even if we do get killed, our brains can be uploaded to computers right away and re-downloaded to clones of ourselves made from our dead tissues. And some people might say, well, is that really you? Do you have a soul? Things like that. Personally, I'd rather have that than be totally non-existent anymore. But there's all kinds of things going on. My number one obsession is not a fear of dying. I mean, I do a lot of dangerous things that people would think are very risky that could kill me. It's not a risk of dying that I'm worried about. It's a fear of missing out on all the great things that are going to happen in the future. People watch my videos, they can see me talk about like making first contact eventually. We're supposed to do it on April 5th, 2063, and according to Star Trek. So I have pictures of them making first contact in Star Trek on some of my slides, and I show myself being suddenly superimposed into that slide. And I say, the reason why I want to cure aging is because when we make first contact, I want to be there. And it's the fear of missing out on all the great things that are going to happen. I'm a scientist, right? I'm interested in all this. I want to know how the universe started. I want to know what the whole structure of an atom is and things like that. And I'm not going to find out if I pass away before somebody else finds it. So that's what it's all about. And I know I went off on another tangent here. We could call this whole series tangents.
But they're really fascinating. And you said something really interesting a minute ago. And for those who do not have a scientific background or technical background, how would you briefly explain oxidative stress and mitochondrial damage?
Oxidative stress is where you produce molecules in your cells that are reactive. They have a charge on them that can bind to other things and actually cause damage. And so you can have like a molecule, superoxide is an example, things like that will actually bind to your DNA and damage your DNA and cause a mutation, and even actually cause the DNA to be broken. And that's one way to actually cause accelerated telomere shortening, okay? Because you can actually cause damage to your telomeres that breaks them, makes them shorter, but it's not in all the cells. It only happens in some of the cells, and those cells can be then replaced by other cells that divide. But that's what oxidative damage is, okay? So mice are constantly being damaged by not environmental molecules as much as molecules they're producing themselves from what are called mitochondria. Mitochondria are the powerhouses of our cells. It's where all the energy comes from. And they're a result of one of those things that I was talking about, horizontal evolution. Okay, it's the term I was looking for. Not horizontal evolution, but horizontal transmission. So mitochondria came from some organism related to a mycobacteria that one day infected a cell, and instead of killing that cell, it became part of that cell. Okay, so now we have mitochondria in all of our cells that came from horizontal transmission, and that mitochondria, it produces energy in the form of ATP, but it also produces these free radicals, these very reactive molecules that cause oxidative stress. But to compensate for that, our mitochondria and our nuclear chromosomes also produce antioxidants, which bind to these free radicals and remove them. Okay, so everything in here's an analogy that's going to be true about everything. We are all completely filled with tug of wars inside of our cells. We have these tug of wars everywhere. Okay, so we didn't evolve free radicals. They just happened. but then your chances of surviving were better if you also evolved some way of fighting these things. So you have a tug of war. Okay. And then we had to evolve something else because we need to, for our species to survive, we have to eliminate the longer lives. So we evolved something else. And and it's not these aren't forces. They just happen accidentally and then get
Yeah.
built into the population. But there's these tug of wars. And so the production of antioxidants and the production of free radicals by mitochondria and the production of antioxidants by mitochondria is a tug of war. And why don't we just eliminate both of them? Well, if we did, we couldn't survive because we have to have some free radicals in order to survive. I mean, there's cells in our body called like macrophages that when they're crawling around inside of our blood, and when they encounter a bacteria that has infected our blood,
Mm.
they shoot those bacteria with like a machine gun. And this is true. Okay. This is crazy. The bullets are free radicals. Okay. So so we need these free radicals to help fight infections. Okay. But then the free radicals have this side effect. They're also causing us to accelerate our aging. So there's got to be a balance. There's always this thing, finding an equilibrium that works the best. And I don't know what the right word is, but I always call it a gamish. Our whole body
Yeah.
is just a gamish of all these tug of wars, things going on that were not designed. They just happen. When something happened, it increased the survival of that individual, which increased the survival of the species, was sustainable. It wasn't retained because that's a force, but it
Yeah.
became sustained because that particular individual helped increase the survival of the species. It's just mind-boggling.
And that's beautifully explained. And it explains a lot of things. For instance, why histamines come into play when we are exposed to allergens, or when we are exposed to strenuous activities or resistance training, the muscles grow because it's a defense mechanism.
Well, the
So
growing of the muscles also accelerates aging of the muscles. Okay.
really,
Anytime there's cell division in the
mm,
human body that's that was not supposed to happen, okay, you
oh,
accelerate the aging process because that causes telomeres. Now, I always
but
say,
that's
what's the
very
point of living a long
paradoxical.
Yeah, I always say, what's the point of living a long time if we're not living? So I don't discourage people from bodybuilding. I think that's really important, okay, for happiness, okay, to make the person enjoy life more, because that's what it's all about. That's one of the reasons I always say that I like the word longer lived instead of old, because life's about living, not getting old.
Wow.
So we got to live. And other things like microdermabrasion, women are, and men too, are killing their skin cells, okay, with lasers and acids and things like that. It's what general term is microdermabrasion. And
Mm.
that makes people look younger because it kills the cells, but it causes other cells to divide to replace those killed cells. And for a short period of time, you look younger because these cells are newly put into the skin, but you've actually accelerated your aging. Well, I'm not going to discourage people from doing that, especially since there's a lot of beautiful women and people out there that, got that way because of it. But it's still, and it makes them happy. Okay. But I always say to myself, I have to hurry up and find a cure for aging, because all these damages that have resulted from increasing cell division is something that I can undo. I can undo the damage caused by that, lengthening telomeres, because that's the main problem. When you induce cell division, the telomeres get shorter, and so you've accelerated your aging. And we just have to lengthen them to undo that problem. And I do strongly believe that I will have the aging process solved. My track record shows it. I mean, my track record,
Mm.
we haven't even talked about that, but all my accomplishments in the past where scientists all over the world failed. And I came through especially when I was told it was impossible, and I got it done. It says that I can cure aging scientifically. The problems are the obstacles. And the main obstacle is the way I can get funding easy. I have no
Mm.
problems finding funding to do my research. I just have problems having control over the research after I get the funding. Okay. And I'll tell you, we'll come back to that. It's just an obstacle that
Yeah.
you wouldn't believe. And it's why cancer is not cured and why heart disease isn't cured and why else.
And
When I know scientists that know exactly how to cure those things. Okay. So, and I do too. Okay.
so let's unpack those obstacles that you're referring to.
Okay. Well, it all comes down to what are the gauges of success in the world? Okay. And people can go to my videos on my website by clicking on that button, key videos and documentaries, and they can hear about me talking about the gauges of success. And so when, like in academic universities and stuff like that, people doing research, when they get a grant to do their research, and I used to be an academic when I was
Mm.
still getting my PhD. I was in a lab where we had to survive by getting grants and things like that. And when we got grants, the whole team got together and said, okay, what can we do to get us more publications, scientific publications? Because your ability to get grants is based on the number of scientific publications you have. The term is called publish or perish. Okay, so are the research labs focused on trying to cure diseases? No, they're focused on trying to get publications. And I'll tell you, every time you publish a paper, an entire team of scientists go months without getting any research done because publishing a paper is so much work. Okay. And so we need to undo that because what the other thing is what's happening is that 99% of all scientific publications are garbage. Okay. Or they're publishing something that everybody already knew. And like scientists that really aren't focused on publishing already knew it, and they would never spend time trying to prove something they already know. So scientists in the academic world will do research to prove something we already know just so they can get a publication, a guaranteed publication, especially if it's already obvious. And so there's a lot of scientists that's all they do is they look for ways to get publications because that's how you get your next grant. Okay, so we have this cycle: get a grant, publish, get a grant, publish, get a grant. Is the gauge of success? Did you cure a disease? No. Okay, and that's a really big problem. I learned that when I was still in graduate school. I was really upset with not just our lack of doing anything when I was in graduate school, but our efforts to try to lie, not me, but lie in like the major professor I was working with, who I'm very fond of, so I don't want to say anything bad about him. But in order to get grants, he had to lie in our grant applications to be able to get grants. And I remember one day thinking, oh my God, our whole lab's going to fall apart because we've been turned down on one grant after another. So he put the word cancer twice on every page throughout the grant application in one case. And I thought, well, that'll never work. But then all of a sudden, I remember the lab assistant, Cindy Borma, came running into the lab screaming, we got it, we got it, we got it. And we got a grant. And all of a sudden, here, everybody's packing their bags, getting ready to find jobs elsewhere or labs to work in elsewhere as a graduate student. And suddenly we realized, oh my God, we're going to make it. And it was all because we lied on grant applications. And that bothered me. I was excited, but it bothered me. And then the first thing that we did was we got into a meeting about publications. Okay, what publications can we do? And so I spent a lot of time just doing experiments to get a publication, and writing the publications was so time consuming, things like that. And I realized what we're publishing isn't really earth shattering. It's not important. The world's not going to change because of this. The only thing that we're going to gain is another
Perspective.
notch on our belt in terms of having a paper published. And so when I finally got my PhD, the last thing I wanted to do was go into academia. In fact, when I got my PhD, I was very interested in finding some way of getting into anti-aging, a cure for aging. I still didn't know about telomere shortening, but I did know about the Hayflick limit. So I did interview, and I applied to a lot of labs throughout the world that were working on anti-aging. And I went to them, and every one of them accepted me. And I would go interview with them to find out if I wanted to accept their offer for me to come work in their lab as a postdoc in an academic environment. And in every case, I thought these guys were totally wrong. I realized we had to focus on the Hayflick limit, not all these other crazy theories and things like that. And so I didn't go into academia. That was the only way I would have gone into academic if I found somebody who really wanted to pursue the cause of the Hayflick limit. But I chose then to go into biotech instead, okay, and work on cancer and heart disease and inflammation and things like that. And we're talking about tug of wars, inflammation. Boy, there's a big one. I mean, there's good and bad about inflammation. It's a big tug of war.
And my question is, how do we break that vicious cycle? Because from the researcher's perspective,
Oh,
I totally understand that they need credentials. They need to publish their work. What do you think? How
you
can we
don't,
break this?
you reminded me of what we were talking. They don't need to publish. Okay. They just need to get grants. And so if we could find some other way to fund academic research, we would have cures for diseases and things like that. And I'm going to come to the solution. I have a solution, but I'll come to it after I explain the next problem. And that's, I didn't stay in academia because of that problem. I went into biotech instead, and I found another big problem. The gauge of success in biotech and commercial stuff is quick return on investment for investors. Okay.
Yeah.
I mean, they all want cures, but they are just driven by this. I want to make a lot of money first. Okay. And so they do whatever they can to make money. And I saw this over and over and over again. The main reason why I left Geron Corporation, which is the place I went to work with Calvin Harley to discover telomerase, is that I was National Inventor of the Year in the United States for my cancer research. So obviously I thought, well, let me see if I can do anything with cancer too. And so I developed a drug which is now called Rytelo, R-Y-T-E-L-O, which is like an inhibitor of telomerase, because in order for a cancer to survive without encountering the Hayflick limit, it has to turn on the telomerase gene. And this is another reason why a lot of people thought telomerase causes cancer, but it has to become a cancer first, and then the cancer turned on the telomerase gene by mutations that allowed it to become immortal, so it could become big enough to kill you. Oh God, I know where it's going. Okay, so the Geron Corporation. So what I did is I developed this antisense oligo called Rytelo. I called it GRN 163. It later became called imeltistat, and then it became called Rytelo. It's just been approved by the FDA last year, and it's actually in use right now for treating cancer. But I'll never forget that, I was like once every 3 months, we'd have big meetings with all the investors. And I would always, because I was the top scientist at Geron Corporation, I would always have to give a presentation. And I pretty much announced, hey, we just cured cancer and aging at the same time, in this probably experiments done the same week. And the investors all were super excited about it. But I remember that the main top investor who worked for a company on Sand Hill in Palo Alto, California, he got up on stage after I got up and he says, okay, we can't focus on two different disease targets. We have cancer and we have heart aging. We got to choose one. And they all chose cancer, even though Geron was short for gerontology, which is the study of aging. They decided there was a quicker return on investment by focusing on cancer. I mean, I'll tell you, I spent a considerable amount of time arguing with them, trying to prove to them they're wrong and that it'll take forever to get this to actually be a cure for cancer. And I said, the number one cause of cancer is aging, so we should focus on aging. But I was outvoted, and we were told to stop all work on aging and focus on cancer. I already spent years. I've already had 6 drugs in clinical trials from previous companies I worked for. I'm very interested in cancer, but I believe that the cure for cancer is curing aging. And so I resigned on that decision, especially when I first started talking about how we were coming up with some products that actually did induce telomerase, and that was like T65, which is now on the market. I was chastised. I was told when I was presenting at a scientific strategy meeting, I was told by actually Calvin Harley. He said, I thought we told you to stop working on aging. I was so stunned by that response that I walked across the hall to my office afterwards, got on the phone and called up some VC friends of mine and said, it's time for me to leave Geron. I need to start my own company and I need your help in doing that. And it was automatic. They jumped all over the opportunity. And so I resigned from Geron Corporation, started Sierra Sciences to totally focus on curing aging. But we have cancer programs too. It's the same cancer programs I wanted to do at Geron Corporation, which Rytelo was only an initial scratching the surface. But that was the problem. The problem was that we stopped curing aging at Geron Corporation because it wasn't the quickest return on investment for investors. And that's the really biggest problem. Okay, so that's the gauge of success there. It's not curing diseases. I mean, we could have had aging, cancer, heart disease, all these things cured years, decades ago, if we would focus on doing that and not on trying to get quick returns on investments. And there's the third category, which is now all these celebrity billionaires starting these companies that are designed to cure aging. And they're not getting anything done. Have you heard anything accomplishments at all? No, they're not. They got all this money, but they're all circuses. They're all, God, I hope I don't get myself into any legal trouble by saying that. But they've hired celebrity advisors, and you know some of them. I even mentioned some of them already. People I know who are more into, they're called redders, people skilled in the art of persuasion. And we haven't talked about this, but I did get a bachelor's degree in psychology too. So I know a lot about things in psychology, and there's a reason for doing that, even though my focus was on aging. But one of the things that I learned about was something called histrionic personality disorder, which is this disorder where people have to have attention all the time. And so there's people that are constantly finding ways to be in the news and things like that, especially in one of the ways is anti-aging field. And so these people become celebrities. There's the redders who are skilled in the art of persuasion. They are good at marketing. And so these celebrity billionaires hired all these people instead of the people that really do the science, that really understand the science. And then these celebrity advisors, they hired celebrity scientists. And these people are sitting around just running around like chickens with their heads cut off and not getting anything done. And plus they're also, because they're more interested in themselves than anything else, they're discrediting all the other approaches that people are doing, like trying to lengthen telomeres, because they want somebody else having all the glory. They want the glory. I'm not in it for the glory. I just want to cure aging, period, and be totally unknown, except for the fact that I want people to know about the struggles I've had to go through. But it's like, that's the third obstacle. I mean, the third gauge of success, reason why cancer is not cured, heart disease, is because people are focused on the celebrities more than they are on the really good scientists that can get things done and know how to do. And there's only a handful of those type of people. And believe me, I know them all. Okay. I'm colleagues with them. I'm friends with we spend time talking about all our same frustrations. We know what to do, but we can't get it done. I have friends that have started companies. The companies failed. No, the company succeeded from the investor's point of view because the investors took over, turned the companies into quick revenue generating projects. When the company succeeded, they sold the companies. The founder of the company, who was a really great scientist, lost everything. They lost all their intellectual property and everything like that because somebody else owned it. Their non-compete clauses prevent them from developing a new company to compete with the companies. The new companies that bought the products, they shelved everything because it competed with something else product they already had on the market. It's just the most ridiculous nonsense thing ever. And people don't know this. People think we haven't cured cancer and heart disease because it's too hard to do, but it's not. We know what to do. It's just that it's not quick returns on investments. And so the solution, this is what I say all this is my call to action. When I speak at medical conferences and stuff like that, I'm saying that the people that we need to reach are these very discreet, ultra high net worth individuals that have more money than they could possibly ever know what to do with. They have no interest in quick returns on investments. They've shielded themselves from the rest of the world on their million-acre properties, things like that. They have hire other people to do all the outside business. They live in their own world. I'm jealous of them beyond belief, but they don't really understand everything. And they think that, they are aging. They wish somebody would cure their aging, but they think it's all being done and nobody's succeeding because nobody knows how to do it.
Mm.
But it's not getting done because of all these gauges of success that are the wrong gauges of success that I was talking about. And so at medical conferences, I say the only people in the world that these very discreet, ultra high net worth individuals have to communicate with are their doctors. Okay. When they have some problem, they can't have somebody else go to the doctor for them. They have to go to the doctor themselves. When they have a loved one that's having a problem, they have to send their loved ones to the doctors and oftentimes go with them. Doctors are the one and only communication, really, between most of these very discreet, ultra high net worth individuals. They're also called shadow billionaires. There's other names for them too. And we need to reach these people. And so I'm telling doctors, when you meet with your very discreet, ultra high net worth individuals, you need to tell them that the system is broken. It's not getting done. It's only going to get done if they come forward. These very discreet, ultra high net worth individuals come forward and start providing funding to the right people, not the celebrities. There's people that have track records like I do. We haven't even talked about my track record, but I have been the reason why many blockbuster biotech drugs have been produced. And it's like when you hear about all these celebrities, I wish people would just say, well, what have you accomplished? And they say, well, I got 50,000 papers published. It's like, that's not an accomplishment. It's like, what diseases did they cure? What diseases did they even treat? And it's none. Okay. But in some cases they have treated something, but it was more, look at all the money we made from developing this product. It doesn't even hardly work at all. I mean, there's a lot of supplements that I take that do benefit me a little bit, but they don't come anywhere close to what I want to succeed at doing. Okay, I don't want to just slow down disease. I want to reverse them, cure them, stop them, everything. And that just takes time and patience. But that said, I'm the fastest at getting anything done anyway, but I still can't get funding. And my friends in the same position, they can't get the funding to do it. And we have to solve that problem. So doctors need to tell their patients that are these very discreet, ultra high net worth individuals that that's what they got to do. And people can go to my website. They can watch me talk about these different things. But we're not going to get anything cured unless we do that. There's none of the other systems. Nothing
Right.
works. Okay.
So let me ask you this hypothetical question. Let's say a genie appeared before you magically and granted you unlimited wishes. What would you ask for? Let's say if it was funding, how much funding would you need and what would you do with that funding?
Well, the more funding, the better. So I wouldn't put a limit on it. But in my business plans, my strategic plans, I call them, I've outlined exactly how we can have aging cured in 3 years with $130 million. I have every penny accounted for. And that's another thing that I am well known for. In every time I've been told to put together a budget and a timeline and things like that, I've always been right on target. People are always raving about how the cost ended up being almost to the penny of what I said it would cost. And it was almost to the day on the day that I said it would be done. So people can actually really take credence in my strategic plan. It's all together. The only thing it's waiting on is the funding. And I can't even hire the right scientists until I have like at least $10 million in the bank that is not being already accounted for, because I oftentimes have $10 million in the bank, but it's already accounted for. But I need it to say, okay, this is money that's going to go directly towards the curing of aging. And then I can start hiring some of the top people, and I know who they are. I'm going to, of course, have a recruiter find people, but I know who I want and bring them in, and we'll get it done. We'll have it done in 3 years with $130 million. Now, that said, that only gets us to the point of where we have a drug that we can add to human cells in a petri dish and show that it completely stops their aging by every mechanism of measurement there is. But we still have to do animal testing. We still have to do clinical testing. And so the average cost of doing a clinical study is 2.6 billion with a B dollars. Okay. So we need to raise a lot more money than just that 130 million. But the way I look at it is when we have a drug that we can drop into a petri dish that stops aging, it's not going to be much trouble to raise more money because we'll have a proof of concept. When this is another silly thing. Everybody's doing anti-aging studies with mice, but mice aging is so different from humans. And I don't understand why everybody doesn't understand that. But I want to do my preclinical studies, so studies with animals for developing a human drug called preclinical studies. I want to do them with pygmy marmosets, okay? Because they're small like mice, their lifespan is short, and we can be doing big studies to show that we can cure aging in pygmy marmosets that would be directly relatable to human aging. Now, that said, the only other non-human primate that is better, shorter lived and smaller is Madame Bertha's mouse lemur, which I used to always say. But then when we started doing studies with cells, we started finding out that, hey, they don't age like humans. They're aging like mice do. Everything is, they're like mice. And so now when I talk about that, I show the evolutionary tree and I show how lemurs are right at the juncture between rodents and humans and primates. And I think they're just misclassified. I think lemurs are actually rodents, but I'm not going to spend time debating on that because I just know that lemurs are not going to work. And it would have been ideal because they are smaller and shorter lived, but than pygmy marmosets. But pygmy marmosets are definitely, they are the right animal. And that's what I want to do. And, but that's, so I would have to, so if I had that genie and I could ask for money, I would be asking for whatever it takes, just unlimited source. That's the number one thing, to get scientists to really focus on curing diseases. You need to make it so that those scientists don't have another worry in the world, okay? They need to wake up in the morning. They need to go to sleep thinking cure for aging. They need to wake up in the morning. They need to dream about curing aging. They need to wake up in the morning curing aging. They need to go to work just thinking about how can we cure aging. If they have to worry about where's the money coming from, what publications do they have to do, what's the investor want, how do I make enough money to pay my bills, things like that, you might as well forget it. Okay, so I've been chastised for this, but one of the things that I always do is I say my number one job is to support my scientists. Okay, I need to make certain that everything in their life is okay. If people say, well, if a scientist is running into financial problems, it's not your job to help them. Well, yeah, it is.
Yeah.
And I do. And a lot of people say, are you nuts? But I do it. And I'll tell you, I developed more loyalty from my scientists than anybody you've ever met. I got to give credit to Dale Carnegie. I mean, back in the late 1980s, I took Dale Carnegie courses on how to win people over to your way of thinking and stuff like that. I was so blown away. I took every course they had. I took every course a second time, and then I took it a third time as a teaching assistant, just because this was one of my arsenals. I talked about this arsenal of tools, but management of employees was one of the most important arsenals. How to get people to really share in your dream. And all the employees that I have had, scientists that I've had working here, have all been obsessed with the same dream. They totally understood my reluctance to work on publishing papers because of the time it took to take up. They all bought into it, and so a lot of them have, because of funding issues, especially because of COVID, they've gone to get jobs elsewhere and stuff like that. I even wrote them recommendation letters to help them because our funding got so low we couldn't do our research. But they all want to come back as soon as I get that funding from some very discreet, ultra high net worth individual that isn't going to try to control the science and, is control freaks.
Yeah.
I have had every billionaire, celebrity billionaire here, names, and you know them. Okay, they've been here. Every one of them wants to have. I don't want to mention that. They want control, and they refuse to sign agreements where I say they can't sell the company without my okay. And they want quick return on investment, even though they already have more money, but they are so brainwashed by their lives. All they think about is, how can I turn my money into more money? And that's why I don't want the celebrity billionaires. I've had a lot of them come to offer me great investments offerings, and I've turned them down just because they won't let me maintain control of the science, and they won't agree to not sell the company. And, from the very beginning, I had no problems finding investors. Okay, so I told you all I had to do was get on a phone call to get the ball rolling. The reason why Sierra Sciences is called Sierra Sciences, the original name of my company when I first founded it was Yonder Technologies, because I think of the yonder years, living longer, want to live longer as being yonder. And so I called it Yonder Technologies. But I went and spoke at one angel meeting called the Sierra Angels in the Sierra Mountains. And I was in line with a bunch of other speakers. We each had 10 minutes to get up there on stage and talk. And one after another, I saw these people in front of me getting up, talking for 10 minutes, leaving with nobody offering to invest, or maybe one person would in the back room would say, let's meet back here and talk. And I was like, oh my God, this is so depressing. What an embarrassing thing to do. And I got up and talked for 10 minutes, and every single person in that room got up, got in line to invest. And so I raised something like $3 million that night. And then it only came more and more. They kept putting more and more money in, but I went from being 100% owner of the company to 17% owner of the company. I had no choices, things like that. They would come in, take over my strategy meetings. I got to admit, there were a few really great investors. And I can't mention their names just because I can't mention the other people's names either. But these people are still loyal to me. They still work with me. I'm still loyal to them, I should say. So I don't want to make it sound like all investors, but they don't have enough money to fund it all by themselves.
They give you autonomy.
Investors take control. They would come in and tell my scientists to start working on other things that have quick return on investments. They would bring in like CEOs that would be more into marketing, creating products. After we succeeded, I absolutely made it clear I did not want to be the CEO. I wanted to be the chief scientific officer. They had to bring in a CEO to run the company. But they brought in all the wrong CEOs that just were devastating. So I fired 4 different CEOs before I finally took over as CEO as myself. We would succeed at doing these quick revenue generating projects, and the scientists would be really upset about it because they were loyal to me. They wanted to cure aging. They loved my passion. They developed the same passion, but we succeeded. And then I remember one day we had this big meeting in this big warehouse conference room kind of thing. And I stood on stage and I explained everything that we had succeeded at doing it to all the investors. And then just like what happened at Geron Corporation, when we had such a meeting, one of the key investors got up on stage and said, wow, Bill Andrews has added so much value to this company. We should sell it. I was devastated. I mean, later I heard friends of mine had the same problems and they lost their companies. But I was going to lose my intellectual property. I was going to not be able to start another company because of non-compete clauses, everything like that. I was devastated. So I was lucky. I had a few of those out of 50 plus investors that were really good that came forward to me and said, let's change things. We orchestrated a whole plan that cost me everything I had to completely take over control of the company again. And those investors stayed with me, but they ended up running into problems with the global financial crisis and things like that, and they couldn't support the funding. And so I ended up buying them out too, but still now I'm very loyal to them. I connect with them all the time. I keep them updated on things. I now own 100% of the company, but I'm never going to allow myself to get back into traditional investors. They're only interested in quick return on investments. And that's why even these billionaires, and when I say a lot of them, I'm talking about 20 different billionaires have been here. I'm right across the street from the airport. They land their 707 jets to 747 jets right across the street with their names across the tops of them, right across the street and walk over. Okay. We have videos of them walking with their entourage right behind them, coming over, meeting with me, and and I turn them down because of their control issues. And right now, I want a cure for aging. My goal is to not make some investor a lot of money. They'll make a lot of money. They'll make gazillions of dollars if they just wait until we actually have a real cure for aging. And when I talk about a real cure for aging, I don't mean something that reverses biomarkers, because that doesn't cure your aging. I call it, what I say, my gauge of success is when somebody passes the Betty White test. Betty White was a friend of the family's. My father was a TV producer, game show producing. Betty White was at our house all the time. She was a good friend. When she started getting into her late 80s and 90s and things like that, she and I talked quite a lot. And I came up with the Betty White test. Okay. And so people can watch my videos, and I show a picture of Betty White when she was 25 and 85. And I say, does anybody here not know which photo of Betty White was taken first? And it says that's what we have to do. We can't gauge our success of curing aging by telomere lengths or DNA methylation patterns or IgG glycosylation or hand grip or things like that. We have to pass Betty. I wanted Betty White to walk out on stage and everybody would see her look and feel and behave like she's 25 again. And though we couldn't measure it, because aging isn't measurable, everybody would know. Okay, I call it qualitatively quantitative. We give it what I call QQ scores. We can't measure it, but that would have been, everybody would have known we had cured aging. Everybody would have wanted to get treated the same way. But that's when I say I want funding to cure aging, I want somebody to pass the Betty White test. And I'm so sorry that it's not going to be Betty White anymore. I still want William Shatner to survive, Dick Van Dyke, and a whole bunch of other people, Sophia Loren, things like that I want them to live forever. I'm so sorry about all the people that passed away. What's his name? God, there's a guy, David Attenborough. He blows me away with his talks. When he passes away, I say he won't pass away if I have anything to do with it. Nobody's going to take the baton. Everybody's going to want and continue his efforts. He wants to rewild the world. Okay, so do I. Okay, but it's not going to happen if he passes away. Okay, and we're going to lose a lot. And every time somebody dies, we lose more information than is in the Library of Congress. That's a quote from Aubrey de Grey. Mm-hmm. But it's a terrible thing. But I want that problem to disappear, period. Okay, right. And so I don't just not biomarkers. I want a cure for real aging. Sorry, sure. Get back on track here.
Yeah. So as it relates to all the work that you and your team have been doing at Sierra Sciences, I believe your team tested thousands of compounds looking for molecules capable of activating telomerase. So what did that discovery process look like behind the scenes?
Well, very frustrating at first, because we first had to design assays, tests. So it's like when assay, a lot of people ask me, what does assay mean? But it means, a test to evaluate whether something is working or not. So we developed a whole bunch of different type of assays. We spent years first thinking we could just really study the dimmer switch, the promoter of the telomerase gene, and identify how it's regulated. But that didn't work. We had a lot of false trails that we weren't down that came up to dead ends. So we finally resorted to, we can't figure out how it gets regulated. And, 30 years later, nobody has. Not a single paper has ever been published or any lab has ever figured out how the telomerase gene is regulated. So we decided, okay, let's not figure out how it's regulated. We'll figure that out later. Let's just find an assay that just tells us if a cell is producing telomerase. So we worked on different assays for doing that. And we learned a lot about it. And there's so many labs in the world that are doing it all wrong. And we've learned how to do it right. We don't publish it because it takes up time, but I do mention it a lot in podcasts and medical conferences and scientific conferences. We just don't publish it. But we went through one revision after another of trying to get an assay that works. And we finally came up with a very brute force assay that worked. Well, we didn't know it worked, but we thought it was the best candidate. We didn't know it worked because we had no positive control to test it with. There was no such thing as a telomerase inducer. Even TA65, which I was one of key inventors of, which is still on the market today, it does not turn on the telomerase gene, okay, at all. It's totally neutral. We use it as our negative control in our studies. But what it does do is it does somehow make any telomerase that you do have a little more active than it was before treating. So it does. So it's still a good thing. It's just not. It's far, far lower in efficacy than what I'm trying to get. Okay, but we had this assay. We didn't have a positive control, and then I'll never forget the day. One of the scientists, Tom Curley is his name. Kerley. He's going to go down as famous in history.
Really?
He comes running into my office, showing me a picture of an radiogram, so we're like exposure of radioisotopes to a piece of film kind of thing, where he had shown that our, so what is it, 314th thousandth, 818th molecule that we tested induced the production of telomerase. Okay, we developed this assay. We had bought these $1 million robots that could test up to 1,000 different chemicals a day, or molecules a day. I don't want to say just chemicals, molecules a day. And it was a very frustrating experience, but
I can imagine.
We were determined. Okay, we kept going and going and going and going. We could test 4,000 a day. So, and we had robots running 24 hours a day. And that one day, and I can't remember the date right now, but I have it in all my plans. He comes into my office, shows me this picture, and I just was in shock. Okay, that we had actually found something for the first time on the history of the planet that anybody had found any kind of molecule that when added to human cells in a petri dish would induce those cells to start producing telomerase. Okay, well, the first thing we did was we sent that molecule to 5 other labs throughout the world and asked them to test it and verify that it works in their hands. And in all cases, all 5 came back and showed data showing that it did work. And they were super excited because they'd never found anything that could do that. And so I remember at a conference in England I was speaking at called the BACR conference. It was my first opportunity to show all that data. And it was like a very exciting presentation because people were stunned that actually there was such a molecule that could work. Of course, at the time, we didn't know how the molecule works because we still didn't know what how the gene was regulated, but it did work. And so here's another tug of war example. Okay, so human cells, they have the tug of war of telomere shortening and lengthening. In our reproductive cells, when our reproductive cells divide, the telomere shorten, but then telomerase re-lengthens. Divided again, telomere shorten, telomerase re-lengthens. So it's a tie in our reproductive cells. So in all the other cells of our body, including human cells grown in a petri dish, it's a one-sided tug of war. We just have shortening. Cell divides, shortens. Cell divides again, it shortens. Now with this new drug called C0314818, because it was our 314,818th molecule we tested, we now could add that to the human cells in a petri dish. It did not win the tug of war, okay? But now it slowed down the tug of war. So shorten, telomerase would lengthen a little bit. Shorten again, telomerase would lengthen a little bit. Short. So this was the first opportunity we ever had for something that could actually slow down the aging And when we do the math on telomere shortening, we know that a human being cannot possibly live longer than 125 years. That's only if they have the perfect genetics and the perfect lifestyle, so they don't have accelerated telomere shortening. But they're still limited to 125 years. And now we have the first opportunity for somebody to actually live healthy for longer than 125 years. Okay, but it's going to take a while. But the problem with that particular drug, because that was a chemical, was that it requires FDA approval. Okay, and so we had to do animal testing and things like that. It was just, and plus it was going to distract us away from doing our other work. So instead of us doing our own preclinical and clinical studies, we licensed it to other companies and told them to do the preclinical and clinical studies, and they could have exclusive marketing rights. Well, they're still working on those. Okay. And I mean, it takes a long time, the average of 12 years to get a drug approved by the FDA. And even to get animal studies done, it's really impossible with the FDA, because the FDA won't approve animal studies, even study aging, because aging is not measurable and they don't consider aging a disease. So lots of obstacles. I agree, aging is not measurable. Okay. But I believe that we could focus on aging-related diseases like Alzheimer's, and then lo and behold, a side effect is that they passed the Betty White test too. Okay, so, but we decided that we better start doing nutraceuticals. So we started testing plant extracts, and we'd already, when I was still at Geron, discovered this TA65 molecule, which we started using as a negative control. Anything weaker than that was considered a negative. But we started testing thousands and thousands of plant extracts. And we were really surprised that none of them worked. We couldn't find anything that worked. And so we were getting pretty frustrated. And then a guy named John Anderson approached us and said that he wanted to have a nutraceutical product that would be a telomerase inducer. And I said, we've already tested thousands and we can't find anything that works. And he says, well, you haven't worked with me yet. You got to work with me. You got to use my plant extracts, and because I know how to make them work. And my scientist thought he was nuts, but lo and behold, he sends us 10 samples.
Wow.
We thought, we've already tested thousands and none of them work. My scientists were just appalled at the fact that we would have to test these 10 samples. But lo and behold, one of them worked. Okay, one of them induced telomerase expression, not as much as C0314818, which actually goes by the name of TAM818, TAM for telomerase activating molecule, TAM818 now. But it was going to slow down that tug of war. It provided the opportunity for somebody to live 125 years or longer than 125 years. We started asking, okay, what the hell happened? And he sent us like 20 more samples. And again, we found another one. And it turned out, it depended on how you made your extracts. Okay. For starters, a plant extract is made up of 200,000 different molecules on average. And people can Google that and find out that that's true. So whenever you have 200,000 different molecules, you're going to find that there's some of them are activating the genes, but there's others that are going to inhibit those activators. And so you again have
It's just isolating the right molecule.
Yeah. So he would fractionate, John Anderson would fractionate the plant extracts into a whole bunch of smaller components based on their size or their solubility or their charge, things like that. So typically, let's say he would send us a banana, plant extract from a banana. He would send us 30 different extracts. Okay, and so we would test these and we found quite a few. And those got combined into a product called isogenesis. So it was first called Product B by a company called Isogenics, and then it got later changed to the name isogenesis. But it was actually a product that was, for the first time ever, turning on the telomerase gene and actually providing some lengthening of telomeres, even though it was still losing the tug of war. But one of the things that we started observing was that there were some people showing some anti-aging effects, especially people that had some severe medical problems in a certain area, or people that were in their 80s and 90s. They were
Mm.
showing seriously unambiguous, even though I'm not prone to placebo effects because of all the clinical studies I've been participating in. I believe these things. I was talking to these people. I was meeting. I was actually seeing for real that there was some anti-aging effects. We couldn't understand why, but then papers came out from other labs showing that telomerase preferentially lengthens the shortest telomeres. And so we started realizing that these anti-aging effects that we might be seeing were due to the fact that these people that were seeing these effects, they had critically short telomeres in some tissues of their body, and those were being lengthened. They were winning the tug of war, even though the longer telomeres were still short, losing the tug of war. So we don't have proof or anything. That's still possible explanation for the results. But then one day we got told it was their number one selling product. We got told they were going to discontinue the product because the profit margin wasn't high enough.
Ah.
Okay. And that was like, oh my God, here's a company who's focused more on profits. Another problem with the gauge's success. I was hoping that working with this company, we would find a nutraceutical that could win this tug of war, but suddenly they lost interest because the profit margin wasn't high enough. So they discontinued the product. Okay, so then years later, we continued testing. Now that we knew that we had to do fractions and there was ways of fractionating, we started working with a company in New Zealand to start creating fractions of nutraceutical. We ended up finding more, and we found a bunch of them that were even more potent than anything in isogenesis. But we were just putting them on the shelf because our goal was to cure aging. Okay, we need to learn from what we discovered to figure out how to find something even more potent. And so we tested 20,000 different plant extracts, which equates to about 600,000 different fractions of plant extracts. And we had found a total of 50 total. That includes the ones working with isogenics. We found a total of 50 that worked, but there were 5 of them that were the top 5 that were more potent than anything in isogenics. And we kept thinking, well, we need to find stuff more potent than that. But then one day I get a call from Touchstone Essentials, a guy named Eddie Stone. Okay. And he runs another company like Isogenics, which was another multi-level marketing company. And he said he wanted to create a product that would lengthen or at least decrease the rate of their shortening and lengthen the shortest telomeres. And I said, other companies have talked to me. They don't want to do it because the profit margin is not high enough. And are you sure you want to? And he said, I don't care about profit margins. I want to make people happier and healthier. That's my goal. That's my mission of Touchstone Essentials. And I'll tell you, that was music to my ears because that's all I was interested in. But I'd never heard of a nutraceutical company or multi-level marketing company or any kind of supplement company that had that kind of mindset. So I said, okay, we'll license you these top 5, but all we want in exchange is a royalty to help fund our research because all this happened after COVID. So we were struggling trying to keep our research going. And so he agreed instantly. And so we licensed it to him. They created a product called Telo-Vital, Telo-Vital. That's now on the market for less than a year now. And it again is showing the same kind of effects that isogenesis did, but it's actually doing better. I mean, I'm so proud of myself for having discovered these things, but at the same time, I didn't stop my research to work on marketing. I let them do all the work and Touchstone Essentials, and they are jumping all over it. Okay, I've become so excited about working with Touchstone Essentials that I now agree to do like videos with them, go to conferences, speak on their behalf, things like that, because I'm thinking this is making people happier and healthier because it's working. Okay, so and I'm not in it for the money. I just want to find a cure for aging, but I think this is a great proof of concept that when more people start taking this thing and start realizing the benefits of it, it's going to make people start saying, hey, let's start making something more potent. And then I'll get funding to continue the research at a level. It costs us, when we have the whole system up and running, it costs us $2 million a month to do our research.
Wow.
And I'll tell you, we don't come up with even a fraction of that now. So our research is at a much slower pace. But I think I think that when the world starts learning more and more about Telo-Vital, we're going to get opportunity to find one of these very discrete ultra high net worth individuals that's going to come forward and say, hey, I want something stronger than Telo-Vital. So I think it's a win-win-win situation for us, Touchstone, and all the people in the world. Okay, so, yeah,
Yeah.
I always forget. How did we get on this tangent?
No,
Okay, so
I think even I forgot how we started and where we ended, but it's all fascinating. And one question, Bill, if telomere shortening contributes to aging, what diseases might be affected by restoring the length? Could telomere biology eventually influence how we treat conditions like cardiovascular diseases, immune decline, or neurodegeneration?
while I was still at Geron Corporation, we did an experiment. Well, we contracted another company to do it for us, but we had this experiment done with baboons, where we would actually look at what's the bifurcation. So when you have your blood vessels going up and down
Yes.
to your legs and stuff like that, just about, just below your stomach is something called a bifurcation, where everything comes together and then separates into your legs. Okay. And there's a lot of turbulence there. Okay. And as a result, there's a lot of wear and tear of endothelial cells that line your blood vessels. And so we are able to measure the wear and tear. And so every time an endothelial cell got worn and torn, it would get replaced by another cell, which would require cell division. And so we predicted that telomere shortening would be faster there than elsewhere in the body. And sure enough, we found this out. We found that and we also found that more plaque was building up right around where there were places where there were endothelial cells and smooth muscle cells, which is another cell that lines the blood vessels, that when the telomeres were short in those cells, there was a higher incidence of plaque being built up. And so we knew from back in the 1990s that if we could find a way of lengthening telomeres, that would be a way of preventing cardiovascular disease, or at least decreasing the rate of cardiovascular disease, because I'm sure telomere shortening isn't the only cause. So, I have a strong background in cardiovascular disease. I'm my track record, my things, I'm one of the inventors of tissue plasminogen activator, which is something still all ambulances carry. When somebody has a heart attack, they inject it into you and it breaks up the blood clots. Millions and millions of people are alive today because of my well, I wasn't the only one, but I was a key person in the invention of tissue plasminogen activator. But the thrombomodulin is another one that's still in clinical studies that actually prevents blood clots. And off on a tangent for quick, the only reason it hasn't been approved is because the FDA said that it would compete with another company that sells a product that prevents blood clots, and that's called low molecular weight heparins. And the FDA is refusing to approve thrombomodulin because they're afraid they'll put that other company out of business. This is the nonsense that exists in the world today that people need to know about. So I know a lot about cardiovascular disease. So I am very, very interested. Anybody listening, the number one doctor anybody should be talking to if they want to fix their cardiovascular problems is a Dr. Amy Doneen in Spokane, Washington. Okay, I think I have her speak at my conferences, things like that. I think she's the world's leading authority on the subject. Off on another tangent, but getting back, I've also have a big career in cancer, and so heart disease, lengthening telomeres should cure all aspects of heart disease, or at least treat them a lot. You have to combine it with good lifestyles and good genetics too. But I can say the same thing about cancer. The number one cause of cancer is mutations. And short telomeres cause mutation rates to skyrocket. And if we could keep telomeres long, mutation rates would be decreased. Plus, the mutation rates also increase the chances that if you even have cancer, first, the mutation rates from short telomeres, when a cell loses growth control, it should be stopped by the Hayflick limit. But the skyrocketing mutation rates, allows some of these cancer cells to start turning on the telomerase gene. Okay, which then allows them to not have telomere shortening, and then they can grow big enough to kill you. And then when they start doing that and you start treating them with chemotherapies and things like that, then they start mutating again to survive whatever you treat them with. And that's why cancers always come back. And plus, the telomeres in your immune cells are shortening, and they lose the ability to fight the cancer. People get cancer 7 times a day. I don't know how many people know
Really?
that. Oh, yeah. And everybody. But our immune system is so good at destroying cancer cells that we never even know it. It's just occasionally one leaks through and then mutates to a way that shields itself from the immune system. And that's becomes a real problem. 95% of the time, it happens because of critically short telomeres causing skyrocketing mutation rates. So if we could find a way to produce telomerase in all of our cells, we could prevent those skyrocketing mutation rates, but we would also keep our immune system strong so that it can fight the cancers. Because one of the main reasons why we lose our battle, another tug of war between cancer, our immune system fighting and not fighting cancer, is because of immune senescence. Our immune system starts fighting the cancer, causes lots of cell division to fight the cancer. Telomeres get shorter because of all the cell division, and then it loses the ability to fight the cancer anymore. If we could keep the telomeres long in our immune cells, it would keep our immune system strong to fight our cancers. I mean, cancer, that's another, I mean, I could say more and more about cancer because I think I already mentioned, I was National Inventor of the Year in the United States for my cancer research. But there's so many different things. Muscular dystrophy, okay, that's caused by a mutation called dystrophin. I mean, a dystrophin mutation in a protein called dystrophin. And people don't know about it when they're young because every time the muscle cells die faster than they're supposed to because of the damage that the dystrophin mutation causes, other cells can divide to replace that damaged cell. Okay? And so when you're short-lived, still raising your young, you don't even know you have muscular dystrophy. Okay? But after you've raised your young, then you start running out of cells to replace the damaged cells, and you start getting the symptoms of muscular dystrophy, and it kills you. Okay? And we never evolved a way not to have that happen because it's better for the species to kill you than it is for you to stay around. But let me talk about every disease. I can come up with an example, but let me give you a simple one. Liver cirrhosis. Okay. Everybody knows that drinking alcohol kills liver cells, but that's okay when you're young because you have other cells that divide to replace those killed liver cells. But when you get in your 40s and 50s, you can't have cells divide anymore because the telomeres have all gotten short from accelerated telomere shortening from the accelerated cell division. And so you just get accumulation of all these dead cells, and that's what liver cirrhosis is, and that kills you. Okay? Again, we never evolved a way not to have that because it's a benefit to the species to get rid of you. But I'll tell you, if we could find a way to lengthen telomeres, it would allow people to drink all they want and never have to worry about liver cirrhosis. Not my goal. Let me one more example. AIDS. Okay? The AIDS, why do people, when they first get infected with AIDS, have no idea that they have the AIDS virus? It's years and years later that they suddenly start getting the symptoms. It's because HIV kills specifically immune cells, especially the CD8 or the CD4. I can't remember which one at the moment, but kills those cells. But other cells divide to replace those killed cells, so you don't even know it. But after a while, telomeres get so short in those cells that can replace those killed immune cells that you develop immune senescence. You no longer your immune cells cannot divide to replace the killed cells. Acquired immune deficiency is why it's called. You've suddenly acquired immune deficiency. You can't divide to replace those killed immune cells anymore because of telomere shortening. If we could find a way to prevent telomere shortening in people that were infected with the HIV, the AIDS virus, I sometimes call it, people could live normal lives with HIV and never even know it, okay? Even though they'd still have the virus, okay? And it's like every disease on the planet. I mean, even the common cold, okay, because especially the effect on the immune system. There's few diseases that I can think of. And in fact, I've only come up with 2 that I know of that probably telomere shortening, lengthening telomeres will not have any impact on. One is sickle cell anemia, okay? I believe that sickle cell anemia has nothing to do with telomere shortening. And maybe I might find out I'm wrong. I hope I do. But The other one is alcoholism. Okay. Alcoholism, I mean, liver cirrhosis is one thing, but addiction to alcohol is nothing to do with telomere shortening. Um, when we talk about, let's go talk to antibodies again, or immune system. Autoimmune diseases, lupus, rheumatoid arthritis, they've all been connected to telomere shortening. Okay. There's this big tug of war between what are called T regulatory cells and autoimmune cells. I forget the exact name. There's a tug of war between these two cells, and it's when the autoreactive immune cells, I think that's the name, start winning the tug of war because of telomere shortening in the T regulatory cells. That's when autoimmune diseases start occurring. And I believe the cure for autoimmune diseases is lengthening telomeres. And there's so many drugs out there that are so devastating for treating people to get rid of like all their B cells that are one type of immune cell. And it causes so many side effects. And that's what's happening right now. We're getting a lot of quick return on investment products to treat people with. The people's lives are horrible after they get treated because they no longer have a functioning immune system without B cells, but it makes their autoimmune diseases go away. It's like a price it's unfortunate. I want a better cure than what we have right now. And I think lengthening telomeres is the best solution for that and every disease on the planet. I mean, at some of the conferences I speak at, I show like 100 different diseases, and I tell the audience I can explain every single one of them in terms of telomeres. And so, yeah,
And
so our world would be happier and healthier if we could
yeah.
lengthen telomeres.
And what would be a realistic future scenario if telomerase therapies became widely available?
All right. Well, one of the biggest problems in the world exists now. Okay. And people don't even know about it. Okay. And that's because we have hospices, assisted living homes, nursing homes cram filled with people that can't take care of themselves. They're depressed. They're miserable. People, this is something. Why don't people know about this? It's because people that are suffering from age-related diseases, I don't, let's say ailments or things like that, they don't want people to know about that because they want people to think they're going to be accepted by society. So people don't go around telling about all the problems that occur when you get longer lived. And so people don't know about this, but this is a really big problem. Ever since I was in high school, I would go to nursing homes because my high school used to put us into a bus and take us there. Kate School is the best high school ever in the world. It's in Carpinteria, California. But they would take us to nursing homes, and we would get off the buses and go in and talk to people living in nursing homes and things like that. Big impact on my life. Most people don't even know about this. I still do this. There's a nursing home here near where I work. There's a friend of mine that's 101 years old. I don't want to say his name, but he's somebody I come in and talk to all the time. And I learn all about the problems and stuff like that associated with aging, but people don't know about it. If 100 years from now, we've had a cure for aging for like 50 years, and I hope it's like 97 years, but we've had a cure for aging and we start seeing the problems that everybody predicts, like overpopulation, the biggest problem. How will the young ever get jobs if the longer live never retire? Things like that. If we have all these problems and somebody says, okay, we need to ban the cure for aging because of these problems, nobody's going to accept that. Nobody's going to vote in favor of that because of the problems that exist now. Nobody wants to go back to where we are now, okay? Because this is the real problem. Humans also have the ability to adapt. I mean, think of before your time, like the introduction of the World Wide Web. When was that? The 90s, I think it was really when it really started taking off. We were all like, oh my God, this is too impossible. We can't survive doing this. This is beyond us. We can't do this. Now we can't live without it, okay? We adapted. We will adapt to whatever. We humans, I have tremendous faith. I don't have all the solutions, but I have tremendous faith that we will come up with solutions for overpopulation, for how will the young ever get jobs if the old never retire, and a host of other things. And my first book has discussions about all those things. But here's a really even bigger problem. There's a talk, several talks. But a key one was one I gave in Tokyo in 2019, or maybe it was 2017. There's something called the silver tsunami, which is, you'd already mentioned lower birth rates and stuff like that.
Yes.
Well, as a result, and that combined with the fact that modern medicine is developing a lot of ways to have people live healthier longer. Okay. We are encountering a situation where we are going to have so many old people, and I rarely use the word old, good for this time, old people that can't take care of themselves, needing to go to assisted living homes, nursing homes, hospices, things like that. And there's not going to be enough people to take care of them. This is called the silver tsunami. The place that's going to be hit the strongest and first is Japan. Okay. And that's why I made a big deal about presenting this at this big conference. I got off the airplane and my picture was on billboards. This conference was so big. And I talked about this upcoming silver tsunami and how the solution was to, instead of making the people get old, let's keep them young. Okay. But another solution is, and I don't really like, this is the fallback solution, is we really need to get all these robotic companies focused on creating robots to take care of to be the caregivers. Okay. Because one of the biggest problems with caregiving is this damage it does to the caregiver. I mean, there are studies, especially published by Dr. Lisa Eppel, who I think I'm a very big fan of. She and others have published studies showing that caregivers are showing tremendous signs of accelerated aging because of the stress that results from taking care of somebody, especially Alzheimer's patients. And so we need to have robotic caregiving where the patient can live with the family, and the family has feeling no burden on themselves because the robot takes care of everything. Okay, that's a priority, but that's until we have a cure for aging, because a cure for aging would be even the better solution. But cure for all diseases, and believe me, when I cure aging, I'm not stopping there. I'm going after everything, every cure that makes life miserable. It's like I want to get rid of all those. But yeah, so every disease, and the world 100 years from now, whenever that's going to be, it's going to be very different, but we will adapt to all the different changes. There's no way we're going to go back to the way things are now.
And I want to spend some minutes talking about your personal longevity practices. And you're also known as an ultra marathon runner who competes in extreme endurance races. So how has studying aging influenced the way you personally live your life?
Well, a lot of different things. Running is one of them. I mean, as I said, I've never in my life liked the idea that I was temporary. I always want to be invincible. I want to find ways to not be one of the victims of all the things that make life less lovable because of this dumb thing called aging and other things. And if people want to know why I call aging dumb, just go watch my videos on why we age. It's not necessary, but it's like, so I've been doing everything. Okay. So part of my efforts to be invincible was, you remember the movie Earthquake that starred Charlton Heston? I mean, it might be before your time. It was, I remember, was that 60s or something like that? I remember watching that and thinking, all these people in Los Angeles or San Francisco or wherever it was, they're all, after the earthquake, their buildings are falling on, killing more people. And I'm saying, why are they all staying there? Why aren't they going off to Nevada or Colorado or something like that and getting away from this earthquake environment? And it's because they weren't healthy enough to do it. They didn't have the means. They couldn't drive cars because the roads were all destroyed and things like that. So I got into horseback riding. Okay, I thought, boy, if ever anything like that happened, I would want to just get on my horse and go there. And so I competed in endurance horseback riding for a long time. I was riding 50-mile races, 100-mile races on horses. And I learned how to survive for both me and my horse. The goal wasn't to win. The goal was to just complete the distance and be healthy afterwards. Okay, and so I did a lot of studies on horses, blood work, things like that, finding out what kind of electrolytes they needed, what caused things, problems, etc. So I learned a lot about health doing that. Then I always realized, boy, riding a horse for 100 miles is a lot of work. And it's like a lot of care for the horse and stuff like that, because the horse doesn't know. The horse just wants to go beat the other horse that's in front of it. And you have to work really hard to restrain, to keep that horse under control. So one day I'm like 30 miles into a training run, and I just completed a big climb up to the top of a big mountain. And I'm sitting there resting my horse, letting him cool down, take his saddle off, let him feed it, get him water, carry water with me, providing him with water and things like that. And lo and behold, a runner comes running up the mountain, this woman, unbelievable. And I said, what? What are you doing here? How did you get here? I mean, how could a human possibly be here? And she says, I'm training for the Western States 100-mile race. And I'd never even heard of that. People running 100 miles. Okay. And I started saying, okay, so what do I do? And so I thought, okay, this is maybe this is a better way than riding a horse. Okay. And so I started talking to some of I never focused on winning, but there were some big, heavy men that were winning some of these endurance horseback rides. And I went to them and I said, how are you doing that when you're so heavy and the horse can't really carry a heavy person that far? They say, well, I get off and run next to the horse. And so next thing I did, I started learning how I trained my horses to respond to voice commands. And I started, I start the race on the back of the horse, but as soon as I got away from civilization, in these races, I would get off and run next to the horse. And next thing I know, I found that it was a lot easier. I got really good at running long distances. I competed in long distance racing in high school and college. I've been running my entire life, but never taken it seriously like I was now. I learned how to survive on my own, how to carry my own backpack. Camelbacks was my favorite, to carry all my water, food, supplies, things like that. And I reached a point where I started saying, I don't need the horse anymore. The horse is a lot of trouble. And so I started competing in ultra marathon running. And lo and behold, my first year of running 50-mile races, I got so addicted to it that I broke the world record for the most 50-mile races completed in a year by a human being. And because I just got addicted, I just couldn't resist. But the rule that I had learned from all my work with the horses, okay, studying the blood work and things like that, is be consistent and keep it fun. Okay. If the horse is having fun, that horse stays healthy. When that horse starts showing signs of, I don't like this anymore, stop, get off your horse, walk it back to where you started from, and let the horse take it over. What you find is that horse gets better and better and better. And so then you reach a point where you can't stop that horse. That horse never gets tired, never gets unhealthy and stuff like that. Super horse. Okay. And so I started doing the same thing with myself. I run every single day. Okay. Tomorrow, just coincidentally, tomorrow, when I complete my 2-mile run minimum, that'll be 6 years of consecutively running every single day without missing a single day. Okay. And so I do that. So minimum's 2 miles a day, but sometimes I run 100 miles. Okay. So but I keep it consistent, and then I also keep it fun. Okay. I mean, there's a few times when I was like 90 miles into an ultra marathon and I kind of thought, hey, it's not fun anymore, but I still finished. And sometimes I'd walk to the finish line, but I wouldn't push it. I didn't mind people seeing me walking, okay, because I knew it wasn't good for me if I started running, because it was causing inflammation and oxidative stress and all these things that cause aging. But it turns out that when you are consistent and keep it fun all the time, you never get inflammation. You never get oxidative stress. You can do your blood markers and you find out you stay healthy. There's people I know that they run every 2 weeks. They go out and run every 2 weeks. And by the way, I focus on running because it's the easiest one to talk about, but kayaking, bicycling, all these different things. There's a whole bunch of endurance sports you can do that are just as good. Okay. And so it doesn't have to be running, but I know people that would like run every 2 weeks and then they'd enter a marathon, and they'd do good, but they'd be finishing on their hands and knees, throwing up, and they would be stiff as a board for 2 weeks because of all the inflammation. Okay. The body thinks all this pain in the joints is something attacking the body. And so your immune cells start migrating, dividing cells, start migrating immune to the joints. The inflammation, the stiffness is caused from all the immune cells that collect there that make it so you can't even bend your joints anymore. Okay. And it's like, this is bad for you. Inflammation, cell division accelerates your aging. And inflammation is the number one cause of aging, without a doubt. And why? It's because it increases your rate of telomere shortening. And but when you run all the day, all the time, and keep it fun, and you're willing to quit an ultra marathon when it quits being fun, except when you're 95 miles into it, you quit and save it for another day, you find out you get better and better. And so, I broke the world record for the most 50-mile races run in a year. And I never, ever had any struggles or problems, never any inflammation or anything like that. I'd get my blood work done. I was always okay. The following year, I broke the world record for the most 100-mile races run in a year. And it was like, I didn't even know that. I mean, it was always fun. I kept saying to people, I hope this never ends. The adventure was just unbelievable. Being out in the middle of nowhere in the wild and seeing all these things and running through the middle of the night with a headlamp. I mean, I loved doing that. Okay. It was fun. I would be finding people lying on the ground and I would help them. I would tell them how to get themselves better. I'd find people that they can't move anymore because their joints were so stiff. I published something called the knee massages, okay, which is some way to restore your knees, because to move all that inflammation out of the way so your joints would bend again. And I would teach people that in the middle of races. I would teach them how to do that, and they'd finish the race. But I ended up getting awards called the Grand Slam of Ultra Marathon Running. I also got another award called, what was that called? Doesn't matter. I didn't even know these awards existed, but I got these awards and stuff like that because I would cross finish lines and it'd be announced that I had just won this award and things. My only goal was keeping healthy, keeping it fun, keeping it consistent. So that's exercise. That's why you can stay young in all different ways imaginable by just exercising correctly. Keep it fun and keep it consistent. But there's also, and I'm heavily into this, the right supplements. Okay, there are supplements that are really, really good for you. The main ones I preach are vitamin D and omega-3 fatty acids, because there's more studies published and peer-reviewed studies with those two than anything else in terms of reducing the rate of telomere shortening. Okay, and there's now some books out written by Dr. Sandra Kaufman called the Kaufman Protocols. And they're the best books ever written on supplements to take. Yeah, and because everybody's different. So you have to, the same supplements aren't right for everybody. So, but the Kaufman Protocol, Sandra Kaufman's a genius. I don't talk about my supplements anymore when I'm on stage. I just tell people to read the Kaufman Protocol. And I tell her that's where I get my supplement choices from, though I'd already kind of came to the right conclusions before that book got published. But I recommend people read the Kaufman Protocols to learn what supplements to take. And there's a test called the ALCAT test that I take every single year. It's a test, ALCAT. You got to say test, because if you search for ALCAT, you're going to get a lot of Alcatraz posts. But you do the search for the ALCAT test, find a place to get that test, because it's the best test on the planet for measuring the induction of inflammation by foods. Okay, so food sensitivity test is called. And it's not an allergy test. An allergy test is using the IgE immune system scenarios. But this is measuring simply the induction of inflammation by certain foods. Take this test. You find out which foods cause inflammation in you. Everybody's different. just quit eating those foods. A year later, you can retake the test and you find out you're no longer sensitive to it, because that's how the immune system changes. So you can start eating those foods again. But I recommend that. Okay, so ALCAT test, taking antioxidants, taking anti-inflammatories, very important. Meditate, yoga. I'm the keynote speaker oftentimes at meditation conferences, yoga and meditation conferences. And I'll show data that people that do yoga and meditation have longer telomeres than their friends their same age that don't. Just because not only oxidative stress and causes accelerated telomere shortening, but psychological stress does too, because that causes all kinds of inflammatory problems and oxidative stress problems. It's important to keep your psychological stress at a minimum. I'm trying to think, obesity, smoking, those things are terrible. They cause all kinds of accelerated telomere shortening. By being obese and smoking, you become more like a rodent, a mouse that has the aging causing problems that rodents do. Okay, so quit smoking, quit being obese, find a way to lose weight. Exercise, best way, be consistent and keep it fun is what I tell people that are obese a lot of times. But there's, of course, I understand that it's more complicated because genetics plays such a big role in obesity. I don't want to talk about the new products that are related later because they have their side effects too. But one of the things I often say when I speak at conferences on running, a lot of these 100-mile races will have a conference beforehand, and I'll be a speaker at that. And I'll talk about how important it is to keep it fun, because running can get boring when you run the same trail over and over and over again. I'll tell people, just follow a coyote. When you see a coyote, run after it. It'll drive it crazy, but you'll go places no human's ever been, except maybe American Indians if you're in America. Elsewhere, you have natives that have lived there longer than you have that have been in those foreign areas, and you get to see those areas. Okay, but that keeps it fun. There's a lot of adventure. Adventure is fun. I'm trying to think of what else.
Sleep?
Oh yeah, sleep's extreme. Proper sleep, like, what do you call it? Sleep apnea. Sleep apnea is terrible. I mean, when you stop breathing, you start generating a lot of free radicals and causing accelerated aging. It's very, very important and inflammation too. It's very important to figure out ways to sleep properly. I sleep with 2 sleep watches every night, and I'm always checking it the next day to verify that my sleep was okay. And if it's not, I go see a sleep doctor. That's another thing. Boy, see your doctors. I mean, there's a lot of doctors that are overwhelmed with the number of patients, and they have to treat patients like cattle because, one patient out, next patient in, next patient out. But there's some good ones, especially concierge doctors that I think is really important to see because they really take a special interest in their patient's health. And well, they all take interest, but at least concierge doctors have the time to. I mentioned Dr. Amy Doneen at the beginning. She's a concierge doctor. That's why she can really spend a lot of time with her patients. And cancer doctors, there's Dr. Jason Williams, who I think is one of the world's top. If you have cancer, first place I'd go is Dr. Jason Williams. Okay, so on diets, like eating the right foods and stuff like that, I recommend Dr. Caldwell Esselstyn. Okay, and Esselstyn. He's written some great books on reducing inflammation from your diet. And like it or not, one of the most inflammatory molecules in our diets is called arachidonic acid. And it's found in fish, meat, fowl, dairy, but not in plants. So that's why I'm a vegan. Okay.
How are you?
I'm a vegan. I live a vegan lifestyle. A lot of people say, "Oh, well, you don't get any protein." Sorry for the dramatics there, but it's like people think plants don't have proteins, but you're right. A lot of plants, if you just ate rice, you're not going to have the right amino acid composition to get the proper type of protein that whey and beef provides us. But there's a lot of great products, and I don't promote products, but I drink what are called only what you need, OWYN, plant-based shakes that have the right amino acid composition. The only reason I do that is because I don't like scooping out powder and blend it in a shaker, but There's even better products that are even better, have better amino acid composition that people don't mind making shakes.
Mm.
The and Touchstone Essentials that I just mentioned, they have like the best shakes, and that's because Eddie Stone wants the best shakes. Okay, the I'm trying to think of but so and the other thing is oils. Okay, so Caldwell Esselstyn has made a big deal about. He's done a lot of studies. He was at the Cleveland Clinic. He's now retired. He did a lot of studies showing that oils actually cause collateral arteries, the microvascular in your heart, to collapse. And so if you have, I have a dog sneezing behind me. If you have cardiovascular, if you're genetically prone to cardiovascular disease, I would say, even if you're not, because, just quit eating oils. Don't even use cooking oil, because oils close up a lot of your arteries. As an ultra marathon runner, one of the reasons I think I do really well is I keep both my coronary arteries and my collateral arteries open, which gives me much more energy for completing ultra marathons. And this is, a lot of people don't know this, and a lot of people don't believe it, but there's really great studies on, when I say collateral arteries, I mean, there are so many collateral arteries in your heart that if you dissolved everything about your heart except for your arteries, you would still see an intact heart, because that's how many collateral arteries are. And they all close up when you eat oils. Okay. Then there's Dr. Floyd Chilton. Okay. He's published a book called Inflammation Nation. That's the best book ever written on the problems of arachidonic acid. Okay. So he preaches the same thing as Caldwell Esselstyn about a vegan diet, to stay away from arachidonic acid. But you also got to stay away from oils. And Dr. Caldwell Esselstyn is one of the few people that actually promote that, because a lot of companies make a lot of profits from people eating oils, and so it's discouraged from promoting that. But what else? So many things that I do. I take sunscreen. Sunscreen is extremely important.
Do you think so?
Take good care of your teeth, because a lot of the bacteria that causes inflammation and causes heart attacks originates from bacteria in your mouth. But there's so many different things, and I talk a lot about it in my videos. Probably enough on that. And
All right. Okay. So, on a closing thought, if we were sitting here again 30 years from now, what breakthroughs do you believe will have fundamentally changed how humans age?
well, if we could overcome this big problem that I talked about with all the improper gauges of success, we could have everything cured. We could, I mean, there are studies that show that if we could eliminate aging, our average lifespan would be 1,000 years. And I show the references a lot of times when I speak at conferences and stuff like that. And people can watch those in my videos. It's like, if we didn't age, and if you're a person like I am that loves living, living's the greatest thing that ever happened to us. I mean, try to think of anything that's happened to us that's better than that. I mean, I think the world would be such a great place. We would also, I believe, if we knew that we could live for thousands of years, we would see wars decrease. We would see terrorism disappear, because nobody wants to put their lives at risk when they have so much to gain by staying alive. Okay. I think it would change only for the good. Okay. Yeah, there might be some problems, but I've faith in humans solving those problems. Even in my first book, one of the problems that I ran into, obstacles, was a lot of religious people would say this is against God's will. God doesn't want us to live forever and stuff like that. So one time I was getting on an airplane, and I was looking for my assigned seat, and I'm sitting right next to this woman who's dressed up in the most elaborate habit that the nuns in the Catholic Church wear habits. She had the most illustrious habit I'd ever seen. And so I sat down next to her, and we got talking, and it turned out she was some high level person in the Catholic religion. And by the way, I don't promote any particular religion. I respect religion a lot. So I believe in all religions. It just happens she was Catholic. But she when she heard that I was working on trying to cure for aging, I had a company totally based on that. She said, "You are doing God's will." I said, "What?"
Wow.
This is so opposite from everybody else. She says, "No, God kicked us out of the Garden of Eden and told us to find our way back. When we were in the Garden of Eden, we were immortal. Afterwards, we weren't immortal, and we're supposed to find our way back to the Garden of Eden and become immortal again and benefit from all the other ways to do it. And you are part of doing that. You are part of God's will." I was so blown away by this discussion that I asked her to write something, and I wanted to include it as a chapter in my first book. And it's in there. So in my first book, there's a chapter written by that nun. I don't know what the correct terminology is. It's really good. So religious obstacles, there's been a lot of those kind of things. And like I remember speaking in India one time, and the audience was appalled at what I was doing. They thought we are supposed to die. And I remember standing on stage and I say, during the Q&A, when so many people were standing up saying that this shouldn't be allowed and stuff like that, I said, I mean, I said, "Do you really want to die so the next generation has a better chance of enjoying life and stuff like that?" And they said, "Yes." And I was stunned by that. Okay. So there are religious cultures, but then I, I meet with people like God, I forget their names, the Dalai Lama. Okay.
Mm.
I haven't met with the Dalai Lama, but one of my colleagues, Elizabeth Parrish, did. She got this one-on-one conversation on film talking with the Dalai Lama. He actually believes in curing aging, which makes you believe that some people that are in a particular religious group, they misunderstand their own religion. They
Yeah.
think that it's wrong, but the top leaders will tell you, no, it's right. It's a mystery as to why a lot of religious groups are opposed to it, but individuals in the religious groups that don't really understand their own religion that are saying this. And I think the religious groups are mostly in favor of curing aging. Yeah, so I think the thing was how will life be different? And there's a lot of ways, all for the good. Nothing.
Right. That was such a fascinating conversation, Per. Thank you so much for being here today.
Thanks for
And
the
there are
opportunity. Thanks for the opportunity. I just love talking about this stuff because I'm just passionate.
it's been such an honor and a pleasure to have you on the show. And I believe there are conversations that inform and there are conversations that challenge the way you see the world. And this was one of those conversations that changed my worldview fundamentally. Because what we explore today isn't just science. It's not just about telomeres or enzymes or even aging itself. It's about question that sits quietly in the background of all our lives. What if the limits we have accepted aren't actually limits? And what if aging, this thing we have been taught to accept as inevitable, isn't a fixed destiny, but a problem waiting to be solved? And if that's true, then this isn't just a scientific pursuit, it's a shift in perspective, a shift in what we believe is possible. Because behind every breakthrough, there's someone willing to challenge the assumption everyone else takes for granted and to ask the uncomfortable question, to go further when most people stop. And today's conversation was a window into that mindset, into a life spent not just studying biology, but questioning one of humanity's oldest certainties. So, thank you so much, not just for your time today, but for your decades of work, for your persistence, and for your willingness to keep pushing on a question that most people wouldn't dare to ask. And perhaps most importantly, thank you for reminding us that the future isn't something we passively wait for. It's something that gets built by people bold enough to rethink the rules. So it's been an absolute pleasure having you on Drug Diaries today. Thank you so much.
Well, thank you. And I think we only scratched the surface. So I look forward to more conversations about this. There's so much. Okay.